The immune system can be divided into a series of functional anatomical compartments, of which the two most important are the peripheral lymphoid system, made up of the conventionally studied spleen and lymph nodes, and the mucosal lymphoid system. Specific homing mechanisms for lymphocytes to each of these compartments serve to maintain a separate population of lymphocytes in each. The mucosal surfaces of the body are highly vulnerable to infection and possess a complex array of innate and adaptive mechanisms of immunity. The adaptive immune system of the mucosa-associated lymphoid tissues differs from that of the rest of the peripheral lymphoid system in several respects. The types and distribution of T cells differ, with significantly greater numbers of gamma delta T cells in the gut mucosa compared with peripheral lymph nodes and blood. The major antibody type secreted across the epithelial cells lining mucosal surfaces is secretory polymeric immunoglobulin A. The mucosal lymphoid system is exposed to a vast array of foreign antigens from foods, from the commensal bacteria of the gut, and from pathogenic microorganisms and parasites. No immune response can normally be detected to food antigens. Indeed, soluble antigens taken by mouth may induce antigen-specific tolerance or antigen-specific suppression. In contrast, pathogenic microorganisms induce strong protective Th1 responses. It is an important challenge to understand these contrasting specific immune responses. The key distinction between tolerance and the development of powerful protective adaptive immune responses is the context in which peptide antigen is presented to T lymphocytes in the mucosal immune system. In the absence of inflammation, presentation of peptide to T cells by MHC molecules on antigen-presenting cells occurs in the absence of co-stimulation. By contrast, pathogenic microorganisms induce inflammatory responses in the tissues, which stimulate the maturation and expression of co-stimulatory molecules on antigen-presenting cells. This form of antigen presentation to T cells favors development of a protective Th1 response. Have a great day.